Welcome everyone, and thank you for joining us for today's session on pioneering our understanding of the human brain. I'd like to introduce Dr. Teresa Cruz. She is a health scientist administrator who manages grants in the Devices and Technology Development and Rehabilitation Diagnostics and Interventions programs at the National Institute for Medical Rehabilitation and Research within the National Institute for Child Health and Human Development. Dr. Cruz will be replacing Dr. Nick Langhals, our original presenter, and begin the track on innovative neurotechnologies. She will be presenting on NIH funding for technology development and translations to humans. My name is Dr. Roshana Kaushanian, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. We would like to extend our special thanks to the Brain Initiative Program Directors, in particular, Dr. James Nadd, for their efforts in organizing today's session. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting your questions during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions in the drop-down box. Our speaker will respond to your questions via email following the presentation. This presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Teresa Cruz.
Hello, thank you for the invitation to speak today about NIH funding opportunities for technology development for translation to humans. Again, my name is Teresa Cruz and I'm a program officer at the NIH. Earlier in this conference, you heard a description of the Brain Initiative from a couple of my colleagues. So I'm not going to talk about the whole of Brain Initiative now, but I would like to bring your attention to our device funding pipeline. At the early stage, we have a new concepts RFA, and this is really for early stage ideas where you don't even have preliminary data yet, but you're looking at developing technologies for invasive recording and stimulation of the central nervous system, and this is an R21. We move down the pipeline to the R01 mechanism, which is what we refer to as our new tools RFA. And this allows for larger applications, larger budgets to really test out those new ideas from the R21 in vivo settings. Following on our new technologies, R01, we have the Brain Device Optimization U01, which is a cooperative agreement. And here we're really thinking about how do we get these new technologies into the hands of the neuroscience community? And what do we need to do to get that technology to be ready for use in labs, or in some cases in human subjects? These three do not require that the ultimate user be a clinical setting, but several of them are. The next part of the pipeline is for human devices. And here we have two different flavors. One is a phased award, and there's two different types here. And then we have a direct clinical trial award. So to zoom in on these a little bit, the purpose here is to support development, optimization, and translational activities and small clinical studies involving therapeutic and diagnostic devices for disorders that affect the nervous system or the musculoskeletal system. So for our phased awards, this is the UG3 and the UH3, or the SBIR U44 Phase 1, Phase 2 Combined Fast Track Award. And for these RFAs, there is some device development that needs to occur, and that's in the either Phase 1 for the Fast Track SBIR, which is for small businesses, or in the UG3 phase, which is for academic institutions. And here you're going to be doing your pre-IDE studies, your enabling work for the FDA. After that's completed and an IRB has been approved, then you can move into the early feasibility studies and our first-in-human clinical trials. It's possible that a technology is ready for that at this point, and in that case, the applicant can go directly into the UH3 Brain Next Generation Devices RFA. So a couple things to note about these funding opportunities for translation. So the first is that they're all cooperative agreements. They are not grant mechanisms. So that means that there is significant involvement on the behalf of NIH staff in the scientific development and execution of these awards. This is particularly related to the milestone development. So much of NIH funding goes to hypothesis-based research or discovery-driven research. These are different in that they are heavily milestoned and they are device development. So there's a bit of a difference here between traditional NIH awards. You need to be working on invasive devices. You need to be tapping into the central nervous system, which also includes the spinal cord and the retina in addition to the brain. These are all considered clinical trials. So there is some intervention on behalf of the device. You need to be prepared with your clinical trial information when you submit to NIH. So you cannot have a delayed onset. And there are some budgetary requirements. These are typically larger budgets, but we do have caps, particularly for the small business awards. We have due dates 3 times a year, and our next due date is in June. So one of the nice things about this translational program is that many of the applicants tap into the NIH Brain Public-Private Partnership Program. This is not required, but we've found that it's been very helpful to many of our applicants. So this PPP program was developed between NIH and several device manufacturers. And the hope was to facilitate partnerships between the individual investigators at academic institutions and the device manufacturers. So in this program, and I encourage you to visit our website, the companies, several of them are listed here, provide technical support and information about their neurostimulation devices. Many of them have capabilities that are beyond what is typically used in the clinic. NIH has also arranged for pre-negotiated agreement templates that can help facilitate those relationships between the investigator and the manufacturer. So each group doesn't have to start from scratch when they work on their intellectual and material property agreements. So now I'm going to give a few examples of grants that have cooperative agreements that have been funded through the translational programs of the Brain Initiative. So the first is from Aisha Gundes and Kelly Foote at the University of Florida. They are working on a closed loop control of essential tremor treatment. This is a deep brain stimulation. And in the video that you'll see in just a couple seconds, you'll see a person who has been who has the device installed, and you'll see that when the stimulator is off, he tremors quite considerably. When the stimulator is turned on, you can see the signal that goes into the DBS system to turn on the stimulator, and that signal is indicative of the beginning of movement. So if you like to play that video now. Okay, so I hope you noticed that this is an example of the public-private partnership between University of Florida and Medtronic for the implanted stimulator. And of course, the benefit of having a closed-loop control where the stimulator is only turned on when the patient is initiating movement is a preservation of battery life and a reduction in side effects from having the DBS on constantly. So another example of the translational program is from Phil Troik, who is the PI as part of a large group out of IIT, and he and his colleagues are working on a visual prosthetic. And in this case, the stimulation is directed onto the visual cortex. So the signal comes in through a camera, it is processed, and then wirelessly transmitted to the microelectrode array, which is installed in the visual cortex. They are working on their pre-IDE work at the moment in an animal model, a canine model, and they hope to recruit 5 human subjects for this study as it proceeds. And our third example is from Greg Worrell at the Mayo Clinic. He is also working in a canine model, but here they are working on an epilepsy therapeutic platform whereby you can measure oncoming seizures with implanted EEG that gets processed, and then stimulation can be administered in a closed loop fashion to reduce the focal epilepsy. And again, we are in canine models, but are planning to move into clinical trials in the upcoming grant periods. I'd just like to take a moment to acknowledge another NIH initiative that is related to device development for stimulation and recording of the central nervous system, and that is the HEAL initiative, which stands for Helping End Addiction Long Term. There are many facets to it, but one of them is this, again, device development pathway, and there are several opportunities for applications to be submitted in response to development of devices for pain management. And I would encourage you to visit the NIH HEAL website to look at those specific funding opportunity announcements. So thank you for your time today and enjoy your conference.
Thank you, Dr. Cruz, for that informational presentation. As a reminder, our speaker will follow up with any questions about her presentation via email. Join us for our next presentation in the Innovative Neurotechnologies track with our presenters, Dr. Michael Bouchon and Dr. John Magnotti, who will present RAVE, R analysis and visualization of intracranial encephalography. Looking forward to seeing you there.